Morning, everyone. I think now it's exactly 11:30. And my name is Luawalo Mofu, and I'm a postdoc at SAIAB, and I'm very excited to have the opportunity to introduce our next guest speaker, Professor Chenidei. Professor Chenidei is a freshwater ecologist. Her interests mostly revolve around conservation and management of riverine and wetland invertebrates. She has supervised more than 20 MSc students and more than 25 PhD students. She co-authored a book on the inland water ecosystems of Southern Africa, and she has published more than 50 scientific papers. And on that final note, she is also a fascinating individual. And on behalf of SAIAB, we are grateful that she's giving a talk today. And without further ado, please join me in welcoming Professor Chenidei.
Where am I? Thank you very much. Good afternoon, everybody. I'm unmuted. Can you all hear me?
Loud and
Hello?
clear.
Great, thanks. Ryan, are you there? Can I just share my screen?
Go ahead, Jenny.
Hold thumbs, it should work. There we go. Right, hello everybody. And thank you all very much for inviting me to give this talk. It's an honor and a pleasure to be able to do so. I think I should start off, since most of you are students here, just explaining how I came to be involved in the management of freshwater ecosystems. It all happened by accident, really. I started off as a marine biologist, and I did my PhD on a group of small crustaceans called Cumacea. Most of you will never have heard of them. But I did that because my children were small at the time, and it was very easy with something taxonomic to be able to put it down and pick it up again. So, but when I'd finished that, I felt that I really would like to do something more live. And I was looking around for something to do, and it just so happened that a friend of mine had been visiting northern Namibia, Jenny Jarvis, who worked on mole rats. And she'd been traveling up through the Namib Desert and had gone past a little pool full of water, which is very unusual then. And the water was full of small crustaceans wiggling around. She went and did her field work and came back a couple of days later, and the pool was completely gone. It was dry as a bone, but there were the remains of a couple of skeletons of these little crustaceans at the bottom of the pond. So she collected the material for me because she knew I liked crustaceans. And so she brought me back a parcel, a little present in the form of a plastic bag with some sand in it. I put that sand into water, and hey presto, within a couple of days, where there was this remarkable assemblage of primitive crustaceans, which completely fascinated me. And that really started me on my freshwater rather than marine journey. But of course, when one's dealing with any freshwaters around here, you all know that they're under threat in one way or another, quite often in more than one way. And that means that we land up very often being involved in management simply because that's the only way that I could see that we might have some very well conserved aquatic ecosystems in the future. So that's how it came to be that I was really involved in managing. Although my passion really is still small hojas in interesting systems, I've spent most of my time on management issues such as water quality, in other words, water chemistry and pollution, invertebrate ecology, mostly in relation to environmental water allocations, and the conservation and management of inland waters. My professional life has consisted, until I retired, of academia, undergrad teaching, postgrad supervision, university and departmental admin. And now I have retired. I've shared most of those academic responsibilities, but I still have a couple of PhD students that I'm supervising at the University of the Western Cape. And I've been involved in a couple of research projects with the University of the Western Cape. I'm also writing and editing various things. At the moment, one of the things I'm doing is revising Vanishing Waters, which is a text that Brian Davies and I wrote, would you believe, nearly 25 years ago. So that will be available online free of charge within the next 6 months, I hope. But that's not to do with what I'm giving this talk about, and that is managing rivers that aren't always rivers. So let's go ahead. First of all, I'm going to say something about what kinds of rivers, of these non-perennial rivers there are. Whoops, why did that happen? Why do we study them? Why is it important that we should look at these rather weird ephemeral systems rather than ordinary perennial rivers? Mention some of their common characteristics, talk about their threats, and then from a management point of view, some of the indicators of condition that we have come up with through the last 3 years of the work that we've done. So let's start off by thinking about the different kinds of non-perennial rivers. First of all, there are those that really are ephemeral. And I should mention that the terminology is very variable. Different people use different words to describe the same things, or the same words to describe different kinds of things. But essentially, I think a useful set of terms is, first of all, ephemeral. Ephemeral means coming and going very quickly. And these systems tend to have very short-lived flow. They're very unpredictable in the volume and timing of water when it does occur. So that, and by the way, this is not a road, this is a dry riverbed, that most of the time is dry, but every so often will become a flash flood, lasting for just a couple of days and then retreating again. So those are the ephemeral systems. Most of them we can think of as being intermittent or episodic, in other words, not always the same. They tend to have very unpredictable flow, and the volumes and timing of water are quite variable as well. This upper one is somewhere in the Free State. This is a rather badly damaged weir on the Berg River. Many of the systems are seasonal. In other words, they're predictable in timing and often in the volumes of water, in other words, rainfall. So for instance, on the right here, this is the Crosamans River, which comes out of the Cederberg and flows towards the sea at Verlorenvlei. And this is another one of these little Cederberg trickles or small rivers that are completely dry in the summer in our case, because we have winter rainfall, but predictably wet in winter. Then there's another category that we're kind of beginning to realize actually does occur, and that is almost perennial. And we discovered an almost perennial system during the work on our non-perennial rivers. Water and therefore flow are present year-round. The volume is usually predictable, even if it's seasonally variable, except when it isn't. And this is a photograph of the same Hjoningnes River, the Navy Yars River that we've been working on. And this yellow stick, where's my pointer, is a gauging rod. You can see that it actually is not even in the water at all, which is extremely unusual. So we were in a way fortunate to be, to be involved in our research at the greatest drought period that we've seen in the Western Cape, probably for 50 years or more. And so this is a perennial river that used to be perennial, but now it's just most of the time perennial. The systems that we worked on, let me just briefly give you an illustration of some of the different kinds of systems. The Navy Yars is the one that I've just talked about. That's on the Agulhas Plain. The Toes River, the system area that we looked at was in this part of the Toes River, which is actually a tributary of the Goerits. The Tankwa River, which is a tributary ultimately of the Doorn, and then the Olifants River. And up here, the Sediments River, which flows into Fleur and I'll show you some photographs of that in a moment. The Tankwa River is the most ephemeral of the systems that we dealt with. These are a couple of photographs taken in 2016. There has been some rain previously, so there are still little puddles left in the riverbed. Most of the time, even those don't exist. But there is a time now and again, which, as I said, these ephemeral systems are very unpredictable. It so happens that in 1992, I was crossing the same river, same bridge over the river, and the Tankwa was behaving like a perfectly ordinary river with water gushing down. But that would only have lasted for a short while, and that sort of degree of flooding is very unpredictable and very episodic. It did, in fact, I've always said that field trips cause rain. And some of you may have found the same thing. You think you're going to be nice and dry, but you go on a field trip and you suddenly get wet. And on this particular occasion, this again is the Tankwa River. This chap here is Pumlani, who had that at that stage spent 2 years working on the groundwater of the Tankwa River, and he had never seen any water in it. It just so happened that we were on a field trip when there had been a bit of rain the day before, and the river was actually flowing. And Pumlani was so excited to see that it wasn't just his imagination that sometimes it would flow. The Toes River is on these photographs were taken very near the R62, where the bridge crosses the Toes River. This is the main stem of the river, again, mostly dry, the remains of a little pond as a result of rainfall, a very rocky bed with very little sediment in it, because the last rains had been sufficient to produce a flood that washed down a lot of the sediment. Upstream is a small tributary called the Prinspoort tributary, much narrower bed, but a couple of pools, at least this one of which is perennial. And I'll show you a photograph of a chap who collects water from there every day for his animals in a moment. The Sediments River, by the way, this is a map which I pinched from Albert. So thank you, Albert. I don't think he knows that I did, but I found it on the internet, and thank you very much. So the area we're talking about is on the west coast, between the Berg and the Olifants rivers, and the river itself is called the Sediments, and it leads into a very, very long and usually quite deep coastal lake called Fleur, which in theory opens at the mouth, but most of the time these days it doesn't, for various reasons, mostly because of abstraction of water. So that was one of the systems that we were going to look at. And in fact, upstream, the Sediments, as I mentioned earlier, is a seasonal stream. And we were hoping to be able to look at some of the features that were characteristic of seasonal systems. A little bit further downstream from where this photograph was taken is a gauging weir. And it's an interesting photograph from 2 points of view. One is the fact that the gauging weir actually extends. We're standing on a bridge looking down on it. The gauging weir actually extends probably for more than 100 meters, which indicates that the engineers who constructed that original gauging weir were expecting that at least at some times the river would be flowing over that entire period. The other interesting thing about it is that the bed has become almost completely choked by reeds, which suggests that there is very little water coming downstream. And when we had a look at Fleur, that became even more obvious. This is an aerial photograph of the Flay itself, which, as I said, is a long coastal lake fed by the Sediments River. These circles are all center pivot irrigation for potatoes, for McDonald's and anybody else who makes lots of potato chips. The Flay used to empty quite frequently into the sea. but it very seldom does now. On the other hand, at the time that we were sampling, when we first started in 2016, the flay down its length was reasonably full, and we thought, that would be great. We will be able to come back and do some sampling here. But from that point on, there was virtually no water in the flay at all. And in fact, we had to abandon those sites completely. And it was only earlier this year, in other words, 5 years later, that there's been any significant inflow into the system. So that's another one of these systems that has changed very considerably from being more or less perennial to being seasonal to being maybe neither. The last of the systems we looked at was the Navy Yars. And in fact, we started to look at it because of another separate Water Research Commission funded project. This is what it's like in its upstream reaches with lots of palmite. Further downstream, it flattens out a little bit. It's a very pretty meandering stream. And eventually, as it goes towards Soetendaal's Flay, which is a big flay on the Agulhas Plain, it straightens out and it has quite a lot of reeds on its banks. So we thought this was a perennial system, and we were looking for a perennial system to do certain amounts of work. And within a year of us starting our non-perennial rivers project, hey presto, there was virtually no flow in the Navy Yars at all, and there was virtually no water at all. So that is our almost perennial system. So why were we interested in intermittent systems? Why was it that we chose to look at them rather than other perennial systems? And there are a number of reasons. First of all, they usually have a lot of subsurface water. This is a photograph, an aerial photograph of the Quiseb River in the Namib Desert. The area here to the south is sand dunes, to the north of gravel plains, and this dark patch here is in fact the Quiseb River itself, and the dark patch consists of trees. So in other words, although there's no surface water for much of the time, there's a lot of underground water, and the presence of trees indicates that. So if we look at a small area in more detail, we can see that, in fact, this is one recruitment of trees, of small, no longer acacia trees, whose name I can't remember. And if you look in amongst the trees, you often find that there are places where animals have been digging. This was a little water hole that had been dug by baboons, and the water was very close below the surface. So in other words, intermittent rivers are often important because although there's no water on the surface, there is water below the ground. And that becomes important both because of the trees, because of the animals that can use it, and also because humans can then exploit it. They also, for the same reason, can act as longitudinal oases. This again is the Quiseb River, a couple of Gemsbok, and a very neat browse line. This is just about as high as they can reach. So they've browsed just about all of the trees that they could. Now, part of the project that we were doing was to look at the way in which these longitudinal oases are used by terrestrial animals. Dr. Mike Grenfell had put in a couple of camera traps along the length of the Toes River, in fact, upstream of the Toes River, but in the Prinspoort area. And he then asked an honors student in biology to go through the images, and she spent hours and hours going through thousands and thousands of images, but some of those images were really very interesting. So these were all taken by the camera traps. That's a little steenbok, blackback jackal, name's gone for the moment, baboons, the inevitable cattle, but even Gemsbok and kudu. So it shows that these systems are extremely important in dry areas, and the drier the area, the more important it is that there are places where there's both food for herbivores and water, and therefore, of course, food for predators as well. The other thing is that there are pools, and the pools in the rivers are actually the most important from a biological or biodiversity point of view. They act as refugia for fish, and there are a few fish in some of these streams, although they nearly always get washed down from upstream. This gentleman is a farmer from the local area, and every day he has to come and fetch water for his cattle from this little stream, from this little pool, which is perennial. There are ephemeral systems like this one, or somewhat more perennial ones like this pool here, but all of them are really important for aquatic biodiversity. Now, in contrast to perennial rivers, which we know something about, it is clear that these non-perennial rivers are actually quite idiosyncratic. It's very difficult to extrapolate from one to another with regard to hydrology or water chemistry or fauna. And that is something we've always been able to rely on when trying to manage perennial rivers. If you know what one is like, you've got a pretty good idea of how some of the others are going to behave. But with these non-perennial systems, that often is not the case. They're also unpredictable hydrologically, so basically stochasticity rules. The timescales of change vary from days to decades. So again, it's very difficult to predict and to see exactly what's going on. They flip-flop, but from being river-like during the times when the river is running, to wetland-like when there's some water but not very much, to dry land-like for much of the time, and then back to river-like again. The two important points that really do characterize these things are that they're greatly dependent on groundwater, and they're very important relationships that exist between flow, sediments, and vegetation. And I want to talk about groundwater and then the relationships in below. And groundwater, if you can imagine, is the groundwater is the water below the surface in spaces between rocks and in sediments. And if this is the surface, sorry, I'm just getting rid of my dogs. Can you shut that door? Sorry about that. Where was I? Yes, so groundwater occurs beneath many of these non-perennial systems. The tree, this is a bad illustration, but the trees would normally have roots that go down to the groundwater, and that is what allows them to survive. And of course, all rivers are fed by groundwater when it isn't raining. It's something that one suddenly thinks, oh, yes, of course, that must be so. But groundwater comes from the uplands and the catchment in most cases, especially where there are very arid rivers. So the groundwater is recharged by rainfall upstream, which means that if dams are put upstream, then a lot of the water that would normally be underground as groundwater in the river simply ceases to exist. And most of the time, groundwater is the only water that's available in these non-perennial systems. So it keeps pools topped up, it supports the trees, and it is depleted by abstraction and upstream dams. Then what about river flow and sediments and plants and the relationships between them? Well, you've seen this photograph before. It's of the Navy Yars River, and the water is flowing. The reeds are on either side, but the flow of river keeps an open channel. But when the flow begins to drop, as it did, you saw this photograph before too, during the drought period, suddenly there isn't a sufficient scouring action and reeds begin to grow. Not necessarily reeds, it can be trees or other bushes, but reeds are the most common ones. So the reeds build up, and there's an equilibrium in most cases between reed encroachment and displacement by floods. And the length of that cycle varies considerably depending on the river, but essentially, as long as the river flows often enough, there is going to be a good equilibrium. What happens when there's less water upstream is that the reeds just go crazy and take over completely. It becomes more and more difficult, and a bigger flood is needed to clear them. And if you see this photograph, it shows a stand of reeds, and you can see the enormous tangle of roots and tubers underneath, which shows why it is that when once they become really established, it's very, very difficult to displace them. The major threat to these rivers, as I've said, is the further loss of water. Climate change obviously has resulted in hotter and drier conditions. Droughts are longer and less predictable. There are more people, but there's less water available. There's more abstraction of groundwater and more upstream dams. So these systems are under more and more threat, although they are crucial for local people. And the methods, as I've mentioned, the methods used for water allocations are specific for perennial rivers. So one of the things we were trying to do was to get an indication of what can we monitor in these systems that will help to track the effects of water loss as a result of management activities. And there are a number of simple indicators which are quite interesting. Just simply a hydrograph, a graph of discharge against time will tell you a lot. This is the Navy Yars River again, 2015, 16, 17, 18, 19, 20. Perfectly normal conditions in 2015, a bit less in 2016, and then from 2017, 2018, 2019, virtually an entire drought. And then in 2020, the flow beginning again. So one can tell quite a lot about what's going on in the river just by looking at the surface water. But of course, that's not the only thing to do, and it's more useful in these non-perennial systems to look at groundwater. And one of the things we can do is to look at depth to groundwater. This is a graph for the SAIAB River in Namibia from 1960 to 2005. And this is don't worry about these other things. This is the groundwater level. The SAIAB River has been used for a long time by particularly the little town of Walvis Bay, and also by various mines. And as a result, the depth to groundwater has continued to decrease, which means that the amount of water that's being used is very considerable. And as the water continues to decline, then water quality often becomes less suitable because you're getting closer to the lower areas of the aquifers. You can also look at simple things like the condition of trees. And if you tag a couple of trees and you can see whether they are continuing to grow or whether they're beginning to decline, that is a very good indication as to what's going on with the water in the river. Another thing is tracking the salinity of groundwater, because as water is pumped out, the salt water would be from the sea, so that's not particularly true for all of the systems we're dealing with. But as long as the fresh water is pumped, is sucked out, so all of the saline water is going to will be sucked towards the pumps. And so if you track the salinity, you have a very good indication as to how much water is actually, how much water is being removed and whether the water that's left is going to be safe to be used. But you can even look at simple things like the growth and distribution of plants. These are two trees, one in the Karoo, one in the Namib, that are on their own in the middle of nowhere. That obviously means there must have been a time when there was sufficient water for them not just to become established, but to grow. They're probably hundreds of years old, particularly this one. But nonetheless, it shows that there has been water available. So if you can plot or track the growth and distributions of plants, you have some very useful information. Even plants within the riverbed. This is the Tankwa River. You can see that there are lots of small plants growing on this side of the riverbed and none on that side, which indicates that when there's flow, it's actually coming round that side, and it will only be very big floods that will remove these plants. So simply knowing something about the distribution of small plants and trees is really helpful. This is another example of the trees from a particular cohort in the NAMIB, and one should be able to track their growth to see how fast they're growing and that they are continuing to receive enough water to be able to survive. So in other words, plants provide really useful information about depth to groundwater and about the rough dates of wet periods when recruitment could have occurred, and the force of the floods, removing plants of differing rooting adherence. Now, of course, all the things that I've said, or almost all of them, are really qualitative. But quantitative data, although they may not be available, it's quite easy to calibrate things like growth of trees and the positions of plants in the riverbed. So it's possible to use all of these to tell us something about what's happening to the water in our non-perennial rivers. The extent and lifespan of pools can also be used in the similar sort of way. But in summary, intermittent rivers, first of all, differ from each other. Very difficult to extrapolate from one to another. They're unpredictable in the amount and timing of water, floods, flow, and groundwater. They alternate between riverine and wetland conditions. They're greatly dependent on groundwater. They're characterized by dynamic relationships between flow, sediments, and vegetation. And we hope that having found some of these indicators, we will be able to put together some good management guidelines, which will help water managers when they start to abstract more water or put up dams on non-perennial rivers, because they're threatened first and foremost by the loss of water to human activities. And I'd like to acknowledge the NPR team, Dominic Mosvamavi, who's the leader, and the staff and students of the Institute for Water Studies at UWC. Thanks very much, everybody. That's it. Shall I maybe you'd like to take this down, or do I take it down myself, or how do I do this? There, it's gone.
Thank you very much, Jenny. That was a very excellent talk. If anyone has a question, please do raise your hand, or just type your comment or your question in the chat box. We still have plus minus 7 minutes for questions.
I see one question here from Josephine Pegg. She says, can you tell me how much of the aquatic life in intermittent pools is left behind from the receding rivers, and how much is opportunistic passers-by? And do you ever find unexpected things in there? Thanks, that's a nice question. Most of the more ephemeral systems will completely wipe away anything that happens to be present. And when the pools are left behind, as the flood recedes, then there are basically two kinds of aquatic. Let's talk about the aquatic invertebrates in particular. There are two types that tend to occur. The one is the fly-ins, mostly insects, well, in fact, all insects, that can fly in to the system, take advantage of the water while it's there, lay their eggs, and in some cases, the eggs are able to, the water lasts for long enough that the eggs can grow and the animals come to maturity. The other major kind are the primitive crustaceans like fairy shrimps and shield shrimps, triops you may have heard of, which survive as desiccated eggs in the sediments. Now, a lot of those get washed away, but I don't think it has been recorded before that in systems like the Tankwa, in the pools, when once the water has receded, there are huge quantities of these anostracans and triops, which is unexpected because one wonders how the eggs could be retained in the system. So with your answer, with the idea of do I ever find unexpected things there? Yes, those primitive crustaceans, the anostracans and tribes, not tadpole shrimps, they're sometimes called. We did not expect to find them in a riverbed in an area where there had been quite significant flooding. So how the eggs survive, we don't know. Thanks. Then there's another question here. Are there any legislative provisions to prevent the damming of non-perennial rivers, e.g. the Assagay River in the Eastern Cape? I don't think that there is anything that specifically deals with non-perennial rivers. So I think that the same legislation, the National Water Act, would apply to them as to perennial rivers. The thing is that we know so little about how to predict the effects of damming. We don't know. With a perennial river, you can pretty much say, if you take off 20% of the flow, then these are likely to be the conditions because the water level will have reached X. And if you take off another 30%, the water level will have reached Y. With these systems, we don't have that kind of quantitative information. But the National Water Act would kick in, and it would be necessary for reserve determinations to be done for that system. But they probably won't be well done in the sense that we don't have enough information really to be able to predict what those effects are going to be. Any other questions?
We still have 3 minutes. Maybe a question from the students would be nice. Okay, just a few rules while we wait for another question. So we're gonna break out from this session at 12:10 to join session 5. If you have any questions or any difficulties, please do type in the chat box and then Ryan will happily assist. So I'll just wait a few more minutes for just one quick question, please.
Albert's asking a question. Hi, Albert.
Hello, Jenny. How are you? Good to see
Well,
you.
and you?
Thanks for
I can't see you.
talk. Yeah. Okay, let me. Yeah, thanks for the exciting talk. Before I started working on fish, the ephemeral rivers were my first love, working in the northern regions in Kariba, Zimbabwe.
Yes. Yeah.
Yeah, it's really nice to see the work that you are doing there. My question, Jenny, is that given the increasing aridity in many of these dryland regions, because from your talk you indicated that these systems, you have a congregation of unique taxa and everything else within a particular region. Has there been any work that has been done to try and document the extent to which these unique taxa are threatened, or to try and predict, because to predict the degree to which the unique fauna of these regions or systems might actually be pushed to extinction? So
I think that the project that we've both been working on, looking at, or that we will be working on, looking at threatened species, red lists and so on, will be really helpful. But by and large, there have been so few studies, even visits to many of these systems, and so seldom does a visit actually coincide with a wet period, that we really don't know very much. We've got, I've had a very good PhD student, James Machingura, who's a Zimbabwean, who's been with us at UWC, and he's been looking at the invertebrates in a number of these systems. So we're beginning to get a reasonably good idea for the systems we've looked at, but more widely, no, it's a big problem. And it's not just the branchiopods, but it's the insects and so on, which I suspect are largely very widespread, but we need to find that out.
Thank you very much, Tanya, yet again. That's the end of this session. But if you feel you have more questions, please do type in the chat box, and Jenny will attend to your questions. And then Ryan, can you kindly open the breakout rooms, please?
Okay, folks, thank you.
Hi, everyone. I'm just going to wait a minute or two to let people decide on where they want to be, and then we can start with the session. My name is Lizanne. I'm a postdoc with SAIAB, so I'll be your chair here. If you do wish to go to the other breakaway room, there should be an option at the bottom of the screen. So this session will be showcasing talks from a number of postdocs, PhD and MSc students, and it focuses on a wide range of topics in both the marine and the freshwater systems, and ranging from taxonomy. We'll have some talks about thermal tolerance, invasive species, parasites, and fish as well. So please remember to keep your video off and to mute your microphone. With regards to the Q&A, the session is a little bit longer than it indicates on the program, so we will go a little bit into lunch. Since people will probably want to go to lunch after the session, I think the best way to move forward here will be to just ask your questions in the chat box for the presenters, and then they can answer there directly. And at the end of the session, if you really have a very burning question that you want to ask someone directly, we can address that. But I think the best will be to just ask your question in the chat box. After the session, we'll break for lunch. You don't have to leave the meeting, just make sure you're muted and your camera's off, and then please rejoin us at 14:30, so that's 2:30 for the rest of the symposium. I think with that, we can jump right in. So our first speaker for this session is Pedro, and he will tell us a bit about work that he did on the taxonomy of fish in South Africa.
Good morning, my name is Pedro Bragança. I'm a SAIAB postdoc, and today I will make a brief presentation on the updates on the taxonomy of the Southern Africa lampies. The Brokerberg family comprise about 100 small-sized fish species, all of them endemic to the African continent, occurring in most of its freshwater environments. The species are popularly known as lampies because of the presence of a bright reflective coloration on the dorsal region of the eye, or Estuaries minors, due to the surface dwelling behavior of most of its species. Recently, the first multi-gene molecular-based study, including most of the family lineages, was published, providing a more comprehensive information on the phylogenetic relationships between the Brokerberg lineages. In the same study, through the inclusion of 2 fossil calibration points, it was possible to have time estimates for the evolution within the Brokerberg, being able to have estimated dates for the origin and diversification of each clade within the family. Another main contribution of this study was in revealing that some genera were paraphyletic or polyphyletic and are still in need of new names. That's exactly the case for the main procotopodid lineage occurring in Southern Africa that I have been calling Southern Africa Lacustricula. As soon as I arrived at SAIAB, I went to the amazing geological collection where I could realize a lot of new species, undocumented diversity, extremely interesting distribution patterns, and also highly informative morphological and osteological characters within the Southern Africa Lacustricula. After just realizing that the Southern Africa Lacustricula was a much more complex and diverse group than I thought, we had to strategize and think on ways of providing quick informations and significant contributions on the taxonomy of the group, especially when we consider this time of biodiversity crisis. So we strategized two ways. One, going through DNA barcoding studies, coupled with in-depth morphological studies of each species, going back to voucher specimens, checking the identifications, and especially providing really precise descriptions of the species. So this is the main thing that we started to think about and strategize. This phylogenetic tree was the first main contribution for the Southern Africa Lacustricula, being possible to first look the relationship between the main lineages of Southern Africa Lacustricula and also the genetic variability within each clade. In the first paper, we decided to redescribe two species. Lacustricula johnsoni, originally described to Malawi, but as confirmed in this study, is a widely distributed species occurring from KZN in South Africa going up to the Congo. And also its sister species, Lacustricula myposei, that have a much narrower distribution occurring in KZN in South Africa and probably in neighboring sites into Mozambique. So we provided precise descriptions of coloration life, coloration of preserved specimens, osteology, including illustrations, and also looking to the pores and cephalic pattern system. Just after the species redescriptions, two new species were described by other researchers, evidencing the importance of species redescriptions for taxonomy, speeding up the process of describing and the discovery of new species. Here, I will highlight a subject that was not really approached on the species redescription paper. Considering that the Lacustriculas in general have about the same size, they are all small species, and in this case, in this picture, we have johnsoni and myposei that are sister species. Why Lacustricula johnsoni have a much broader distribution in the gray color in the map than myposei, that is in brown, occurring only in KZN and probably in their neighboring areas in Mozambique. So this is probably due to a difference in ecology or to a historical process. I have a similar question for the sister species, Lacustricula catangai in red in the map and Lacustricula medulateralis in orange. Which are the main things affecting their actual distributions? Early this year, we published a second paper on the Southern Africa Lacustricula, focusing on the Lacustricula huterari species complex that represents the ancient lineage to diverge within the Lacustricula, the Southern Africa Lacustricula group. Okay? In this paper, we revalidate and redescribe Lacustricula chabensis that have been synonymized with Lacustricula huterari, a species originally described from the Congo, from the northern savannas of Congo. And we also describe a miniature species called Lacustricula pygmeus. This study included only morphological characters. And even it was able to find several informative and interesting characters in distinguishing the species within the Lacustricula huterari complex. The osteological information also proved to be extremely important and informative in distinguishing the species within the Lacustricula huterari group. Here is a preliminary tree of the Lacustricula huterari group, showing first the relationships between the species, a major genetic divergence between Lacustricula huterari from Dungu Congo from the remaining species, and also probably indicating that there is still a new species to be described, as suggested by previous examination of morphological specimens in the collection. Okay, but what about the other Southern Africa Lacustricula species complexes? Ongoing studies are indicating that in other regions in Southern Africa, we have a similar pattern of extremely complex cases where we have 3, even 4 species occurring in the same place, in the same locality. And historically, they have been cataloged under the same name. So in this case, these fishes occur in the Okavango. They are not all the species occurring in the Okavango, but the species that have been historically classified, cataloged as Lacustricula johnsoni. That is the top fish in the picture. So the other 3 fishes, we need to check if there are any available names, and also probably they all are new species. So we are in the process of describing them and checking if there is any available name. The same case we're doing, same similar study we're doing with the Cunani fishes. So everywhere you look more in deep, you look that there's a lot of new species to be described and really interesting things to investigate. Which are the impacts of this research? Certainly the main impact is on conservation. Now we have better delimited species. We know that there are many species that still need to be described, and we have a better knowledge on the species distribution. This is basic information for conservation. Also, studying this group, I believe that we'll be able to improve our knowledge on landscape evolution through biogeographical studies. The ecology and morphofunctionality questions come more and more each time. How is it possible to have 7 or 8 species in the Okavango, for example, occurring in close areas, even overlapping their distribution? How is that possible? On the health side too, there are any of these species, they have a preference to eat like mosquito larvae. Maybe that can be a really important information for the future measure and try to combat mosquitoes. Also on the economy side, in many places in Africa, for example, in Lake Malawi, Lake Tanganyika, in the Congo, Nigeria, there are really well-developed export industries of aquarium fishes. Why that doesn't happen really in Southern Africa? We have beautiful fishes. These fishes, when they reach the main aquarium trades and aquarium shops in Europe, they cost like 10 euros, 20 euros each. So it's much more valuable than only selling fish to eat. So maybe this can be a way to improve the income of many poor fishery communities and boost their income. So that can be really helpful. At last, I would like to thank NRF SAIAB, especially the staff at SAIAB that makes this work possible, and especially the collection team and the molecular lab platform team. I'm also grateful to Albert Chakana, who is my supervisor, and I really enjoy having fish talks with him and learn a lot from Southern African fishes with him. I'm also grateful to the other SAIAB freshwater fish taxonomists and to my collaborators from Belgium. Thank you a lot. I hope you enjoy the talk. And if you want to contact me, please just send an email. Thank you.
All right. Thank you very much, Pedro. I've been learning quite a bit about the hidden diversity that we actually have in South Africa today. So thank you. Our next speaker will be Josie Pegg, and she will be telling us about carp from the Orange River. So thanks, Josie.
Hi, I'm Josie Pegg, and I would like to tell you about the Orange River carp hunt. Like all good adventures, I didn't go alone, and I'd like to acknowledge and thank everyone who helped me catch fish on this survey. What was the purpose? The mission was simple: to find carp. Carp are one of the world's most problematic invasive fishes, but whilst much is known about their impacts and ecologies in still water systems, much less is known about what they do in rivers, and I was looking to find that out. I was working on the Orange River. This is a terrific study site because it covers a huge distance from the cold Lesotho mountains to the warm oceanic waters on the borders with Namibia. Also along this river, there are both sort of fairly natural river sites, but also a number of significant dams and natural barriers. So there's a lot of different environmental variables that we could look at the natural and invasive biodiversity. At each site, I used a number of different methodologies to capture fish. I used gill nets, fyke nets, short seine nets. I used electrofishing and rod and line capture, as well as sort of manual searching using a kick net. All the fish captured were measured, identified, and a tissue sample for isotope analysis. And I'll explain a little bit more about that shortly. But let's get going on the survey. Our first site was on the Lesotho border. There's the Malotti Mountains just on the horizon. Up here, a lot of the river was very narrow, but this was my survey site, a very typical stretch. A real feature of the river here is that the flow was very low and very unnatural. There are huge dams in Lesotho, and releases of the dams are really what drives the flow regime here. You can see there's a combination of scoured rock and settled mud, and no in-water plants or significant structure. So the biodiversity was very low, although we did find some lovely juvenile yellowfish and carp. The farm we were staying at had rock art in its caves. We had to sort of climb down a little cliff to get to these, which wasn't in my risk assessment, but I totally think it was worth it. They were spectacular. From Lesotho, we headed downstream to Budfontein. This is just upstream of Hariep and downstream of Aliwal North. Here also, the water levels were very, very low. Angelica is an average-sized lady, and it only came up to her shins. But we still found loads of carp, and talking to the farmer whose land we were working from, he said that there had been fish kills of native yellowfish and mudfish, but in the pools separated from the river, there were still large numbers of carp and barbel. There was also, when we were working on this site, a unscheduled dam release, which really changed the nature of the water. The river level was much higher, much faster, much dirtier and foamier. I lost 2 fyke nets, which I had to fess up to Olaf, but it also was like a real good demonstration of how extreme the conditions are for fishes that have to live here. So, lots of carp and not a lot else. Our next site downstream was Hariep Dam. I like this picture because it shows me using a clean pair of scissors to take a small fin clip. When we do stable isotope analysis, we use tissues from fishes as well as plants and invertebrates to construct food webs. Looking at the ratios of carbon and nitrogen allows us to look at what something has eaten or been eaten by, and also examine the trophic niche of species. This is useful when you're considering invasion biology, because by comparing the trophic niche of your invader to your native species, you can see if there's overlap or predation or displacement. Here we are collecting the putative items. And here's one of those big Hariep carp. Our next site was Flashbank, between Hariep and Vanderkloof. Again, this was a fairly natural riverine stretch. There's Olaf using his rod to sample for fish for me. And there's the rest of us sampling waist deep in the slippery waters. Now, the fish were also quite slippery here. but they were slightly different to what we'd seen previously. Here's a southern mouth brooder. These are found downstream of Harib, but not in Harib or upstream. And what you'll see as we go on is that biodiversity increases little by little as we move further downstream. We also found a rock catfish. This was actually found dead. And I've included it because although we were using a really comprehensive survey methodology, this is really an acknowledgement that no matter how good your sort of broad biodiversity survey is, you're still going to miss some species. This one was only recorded because it was found dead. It wouldn't have been captured otherwise. So it's sort of an acknowledgement of the fact that this is still an imperfect survey, although comprehensive. So the species list grows a little. Between Flashbank and our next site, it rained a lot. One of our windscreen wipers flew off going down the N1, which led for a few seconds of excitement as I couldn't see out the windscreen. But we lived and made it to Philippoulis. We were staying in Philippoulis because Site 5 is Vanderkloof Dam. Although very large, Vanderkloof doesn't have particularly a lot of features. We found loads of little barbel. We saw some really cool terrestrial wildlife. We were actually working on the dam through a private game reserve, so there was some nice animals to see on the drive in. But we also found carp, as well as small and largemouth yellowfish, mudfish, barbel, and the southern mouth brooder. From Vanderkloof, we crossed the border into Prieska, the Northern Cape. I love Prieska. It is a really great little town. I like it for a couple of reasons. Firstly, because it has this absolutely beautiful little church, which I just love. It's my favorite in South Africa. But also because in Prieska, you can buy anything you might possibly need on a field survey. And the reason for this is that Prieska, although small, serves a huge farming community. Although the area is very dry and arid and northern capey, along the Orange River, there is a huge amount of agriculture, and it really shows, just as Prieska is sort of the heart of the people, the Orange River is the sort of heart of agriculture and very, very productive. Here's Spa and Nawa electrofishing, and here's Nabukle relaxing with her rod and line. Certain slightly cynical people have suggested that the Orange River carp hunt was just a big fishing trip, which I think is really rude. It was a very hard working survey, but we also did do a lot of fishing, and there was a good reason for this. Carp are actually a species that's quite hard to catch using nets. They're quite savvy. They tend to jump the nets. They're also quite difficult to catch using electrofishing because they're such big fish, they feel it from far away, and it's quite difficult to get them without them escaping. However, they're greedy fish, and they are not too difficult to catch using rod and line if you know what you're doing, as Nabukle does. So we continue. As we're in the Northern Cape, the species list has changed a little. We've added the Mozambique tilapia and mosquito fish, but the carp are still there. Our next site, Site 7, is Brokerberg Dam. You can see here, unlike a riverine site, there's this real big tranquil dam. Again, very productive, lots and lots of flies, as shown on this image. So many flies, in fact, that poor Spa slept in the car one night because his room was infested. But he didn't seem to mind. Look at him. But I like this picture because it shows not only a happy Spa, but also the difference between the unnatural dammed environment and the natural riverine environment. But we caught carp within the dam. Here's one of our fishes. We also caught our first little barb, a straight fin. Our next stop was downstream of Ochrabes. Like the dams, Ochrabes is a huge natural barrier to upstream migration, but this is a natural barrier as opposed to a dam. We're also in sort of the real Kalahari, hot, dry type environment, although again, alongside the river, very lush, very productive. Nabukle caught a little barbel. A few days later, somebody in exactly the same spot, check out the hills behind, caught a much bigger one. But size doesn't matter. That wasn't the point of this survey. Our species list grew a little further, but still we were finding the carp. Our penultimate site was the Ice Richtersveld National Park. Here we were really in the wilds. It's a spectacular place if you have opportunity to visit. The landscape's beautiful, the rivers are beautiful, the skies are beautiful. And we also found some really cool fish, like this little 3-spot barb. But we also found loads of carp. And I thought it was quite interesting here in this sort of grassy, shallow environment, which is actually perfect carp territory. There was loads of carp, like literally 100 carp swimming around here. And it really makes you think about how differently aquatic invasives are dealt with to terrestrial invasives. Within a national park, would they allow, 100 non-native mammals to wander around? I suspect not. And yet here were these carps freely living their best lives. There's one of them. So our species list continues to grow, but carp ever present. Oh, this is just another little aside. This is called the Hand of God. It's a natural structure. It's not a carving that's present within the Eisrichtersveld Park. Nothing to do with fish, but still quite cool. We liked it. We hope you do too. So, on to our final site, Beauvallon, the tidal limits of the Orange. You can see here that the plant life is becoming much more sort of salt marshy, and this sort of tidal ingress was reflected in the fishes. For example, here's a mullet. We also got some freshwater sardines, but yes, carp still present. They didn't mind the salt. This was the site with our highest number of species, both the sort of freshwater species and the sort of brackish species. So as the sun set on Namibia and we neared the end of our Orange River adventure, we kind of felt it'd be wrong to come all this way without actually getting to the sea. Here we are in the estuary at Oronyamund. Now we're from landlocked Zambia, had never seen an estuary before. He quite liked it. We dipped our toes in the cold Atlantic waters, and then we had to head back. It was a bit of an epic drive because this was the 17th of March 2020, just before we went into hard lockdown for the first time. So what can I tell you that we learned about carp on the Orange River? Well, carp were present at every single site we visited and were relatively easy to find. What was quite interesting was that downstream of Haripe, all we found was adults, and upstream only juveniles. I honestly believe this is probably more of an artifact of the survey methodology. I would be amazed if carp weren't breeding downstream. But it really does ask questions about how much sort of gene flow is there from the sort of higher waters downstream. Are carp moving downstream? Are they being spread? We've taken genetic samples, so hopefully we'll be able to answer that. Whether there really is distinct populations, there are some really significant barriers, but maybe fish are moving downstream. Hopefully we can find that out using genetic material. And then carp versus biodiversity. If I'm honest, I really saw no signs that carp were harming the native wildlife, but they were very much present at the sites where natural biodiversity was low. Now, my feeling is that that is simply because up in those upper reaches of the Orange, where there was a weird regulated water flow, the conditions were very extreme, and carp were able to sort of occupy a free niche and tolerate much more than the native fishes. But once I've got the isotope information back, I'll be able to look at whether it appears they are, overlapping in their diet or competing with native fishes, or whether they're just sort of occupying a space that's been freed by other anthropogenic impacts. So as that story ends, and we're all hopefully looking forward to a future where we get back on the road, I hope a few things for you all. Firstly, that you find some, weird and wonderful places along the way to stop and grab a ginger beer. I really hope that you find the dead frogs in the swimming pool before you swim and not after. But most of all, I hope you find really great teammates to catch lots of fish with. Thank you so much for listening. I hope you enjoyed our adventures on the Orange, and I am more than happy to take any questions. Thank you. Goodbye.
Oh, great. Thank you so much, Josie. That's super interesting. Your trip sounds like it was really, really enjoyable. Our next speaker is Kerry Ann, and she's going to be telling us about the thermal tolerance of aquatic biota in urban estuaries.
Good day all. I will be presenting my postdoc research titled Evaluating the Thermal Tolerance and Performance of Early Life Stages of Fish and Invertebrates in an Increasing Temperature Variable Warm-Temperate Urban Coastal System. Within the coastal realm, estuaries are fundamental for biodiversity, mostly acting as nursery and settlement areas for intertidal or transient larvae and juveniles of marine fish and invertebrates, for example crabs. Urbanization, as well as rising mean ocean temperatures and local temperature variability, however, may be placing ecological and physiological pressure on these early life stages, recruiting into these urban coastal systems. For instance, within the larval life stages of crabs, which usually comprise of zooal and megalopal stages, thermal tolerance windows for optimal daily functions such as breathing and feeding may be narrower for the megalopal stage compared to the zooal stage under increasing or variable temperatures. This may be due to the zooal stage having less energy demands compared to the more developed megalopal stage as a result of the surface area to volume ratio of respiratory organs decreasing as ontogeny progresses. There is therefore an urgent need to produce reliable knowledge of an organism's physiological response to anthropogenic mediated changes within coastal urban habitats, especially for early life stages of invertebrates due to very little literature available, in order to assess the vulnerability of each life stage and ultimately communities. For this study, the aim was to evaluate the thermal performance of early life stages of vertebrates and invertebrates collected from artificial seawall and jetty habitats within the urban Port Alfred coastal system, situated along the warm-temperate southeast coast of South Africa. This was done by achieving three objectives. Firstly, by determining the upper and lower thermal limits of early life stages of selected fish and invertebrates using the static or incipient lethal temperature method, at which 50% of a sample die when subjected to an acute increase in temperature. Secondly, by determining routine metabolic rate, maximum metabolic rate, and metabolic scope of early life stages of selected fish species using static respirometry and pyroscience equipment at various test temperatures. And thirdly, determining oxygen consumption rates of early life stages of selected brachyurans using closed respirometry and pre-sense equipment at various test temperatures. For the purpose of this presentation, the focus will be on preliminary results and methods for Objective 3. Brachyuran larvae were collected from two artificial microhabitats, jetties and seawalls, within the urban Port Alfred coastal system, situated along the warm-temperate southeast coast of South Africa, using light traps deployed for approximately 12 hours overnight and retrieved the following day at dawn. After collection, larvae were transported back to the Aquatic Ecophysiology Research Platform Laboratory in Makanda in aerated 5-liter containers containing estuarine water of the same temperature at which they were collected. On arrival at the laboratory, each of the 4 site samples was sifted through a 65 micrometer mesh sieve before being placed into separate aerated 500 milliliter water beakers, filled with filtered seawater and acclimated in a water bath for at least 1 hour at the temperature in which they were collected. After the acclimation period, brachyuran larvae were coarsely sorted into dominant selected species of Zooea or Megalope before being placed back into the water bath. Dominant selected species included Pinotherus species and Hymenosoma operculata. Water temperature was then ramped up or down by 1 degree every 15 minutes until the desired experimental test temperature. Test temperatures used included 11, 15, 19, 22, and 25 degrees Celsius. 11, 19, and 25 degrees Celsius represented the lowest nominal temperature, the average temperature, and the highest rounded off temperature from hourly in situ environmental data collected during 2019 and 2020. Oxygen consumption rates were measured using an optical fluorescence-based oxygen meter. A closed respiratory system was utilized where up to 4 larvae, depending on their size and life stage, were placed randomly in 18 individual sealed 80 microliter or 200 microliter wells containing filtered seawater within a Loligo system 24-well glass microplate. Each well on the microplate contained a non-invasive sensor spot affixed to the bottom, which are read through the transparent microplate by an optical fluorescence oxygen 24-channel SDR connected to a laptop running the SDR software. The remaining random 6 wells were left blank, containing filtered seawater serving as a control for background respiration. Measurements of oxygen consumption were taken every minute for approximately 60 minutes. Only the first 30% in linear decrease in air saturation was used to calculate the oxygen consumption rate. At the end of each oxygen experimental trial, larvae were preserved separately in epidorfs containing 99% ethanol for photographs and further measurements to determine individual mass. For both species at the zooea and megalopal stage, a total of 3 replicate trials for each test temperature was conducted. For the statistical analyses, generalized linear models will be carried out on the zooal and megalopal life stages separately to determine if log-transformed mass-specific metabolic rates vary between sites, test temperatures, and species. Metabolic scaling coefficient will then be calculated for both species at both life stages using the power function. Linear regressions will then be calculated from an Arrhenius plot on mass-corrected respiration. And for all statistical analyses, assumptions of normality and homogeneity of residuals will be tested using Shapiro-Wilk and Levine's test respectively. Preliminary oxygen consumption rate results, which have not been corrected according to individual masses yet, with the first 15 minutes excluded, overall indicated that oxygen consumed by treatment samples, which are highlighted in black, decreased with an increase in temperature for both JTB and D across the zooea and megalope life stages of Pinotherus species, compared to the control samples, which are highlighted in grey. The megalopal stage of Pinotherus species, highlighted in green, however, had lower oxygen consumption rates compared to the zooal life stage, highlighted in purple, especially at higher test temperatures of 22 degrees and 25 degrees. In addition, from the graphs observed, the 15-degree test temperature may be preferred or optimal for both life stages of Pinotherus species, due to oxygen not being consumed excessively for the treatment samples in black and being close to the control samples in grey. Furthermore, for the zooal life stage across all test temperatures, there was no significant difference in oxygen consumption rate for treatments between JTB and D. For the megalopa life stage, however, at the 25-degree test temperature, there was a significant difference in oxygen consumption rate for treatments between JTB and D. These preliminary results suggest that the megalopal life stage of the brachyuran pea crab Pinotherus species may be more vulnerable to increases in temperature above 22 degrees compared to the zooal stage. This may be a result of crab larvae becoming increasingly xenothermic as they develop, which may make them vulnerable to increases in mean ocean temperature and temperature variable events such as marine heat waves, which are expected to increase in frequency and intensity in this region. Furthermore, it may create a potential bottleneck for successful recruitment of crabs in the megalopal stage, especially in artificial habitats such as seawalls and jetties, which already lack complexity for refuge. Going forward, respirometry trials for Pinotherus species and Hymenosoma operculara larvae for both life stages at 5 test temperatures from jetty and seawall artificial habitats in Port Alfred need to be completed. Routine metabolic rates, maximum metabolic rates, and metabolic scope of early life stages of selected fish species using static respirometry and pyroscience equipment at 5 test temperatures still needs to be conducted. And furthermore, determining the upper and lower thermal limits of early life stages of selected fish and invertebrates using the static or incipient lethal temperatures method also needs to be conducted. Finally, comparing the metabolic rates for both these fish and invertebrate selected species at different artificial habitats across life stages to in situ environmental temperature data collected to determine if they are vulnerable in this warm temperate region of South Africa needs to be conducted. Potentially, conservation measures for early life stages in urban coastal systems can then be developed. Thank you so much for listening.
Cool. Thank you very much, Kerry. Next is my own presentation. So I hope you find it interesting. And please remember to ask any questions in the chat box if you have any.
Hello everyone, my name is Lizonda Necker and today I'm going to be telling you a bit about work that I did in my PhD that looked at the resilience of an aquatic invertebrate community in a naturally saline lake during a supra-seasonal drought. This work was published this year in the journal Water, so if you would like to know a bit more about what we found at the end of the talk, please feel free to have a look at the paper. Natural saline lakes are found all across the world, making up 44% of the global total lake volume. They are most prominent in arid and semi-arid regions and range from large permanent lakes such as the Caspian Sea to small seasonally filled lakes or ponds present across many regions including Australia and Southern Africa. They are different from marine environments in the sense that they have no physical connection to any marine habitats, and they have a much more pronounced fluctuation in their salinity compared to the stable salinity of the ocean, for example. These fluxes in salinity occur naturally and usually on a seasonal or annual basis through the introduction of fresh water. These changes in salinity also alters the aquatic environment and thus also the aquatic community structure. Generally speaking, you will find that there are more freshwater or saline tolerant taxa during the wet season when there is a decreased salinity, while in the dry season, when salinity is slightly increased, the biota will be more saline adapted. This creates a highly unique environment with distinct aquatic biota. Saline lake ecosystems therefore often have quite a high environmental and conservation value. Unfortunately, this value is often overlooked because it is difficult to put the ecologic value into a cash value. Climate change, with its associated changes in temperature and rainfall, and anthropogenic alterations to the inflow of water through damming and water obstruction, are the two primary threats that saline ecosystems face. These habitats have a very specific hydrologic budget that the biota have adapted to, and a change in this will modify the salinity and prevent the natural cycling in salinity, which can eventually lead to a loss of biodiversity. The worst-case scenario for these systems is a reduction of water inflow that can cause severe salinization or increased salinity and may eventually lead to the total desiccation of a saline lake. Harsh and long droughts in arid and semi-arid areas such as Southern Africa are often triggered by strong El Niño Southern Oscillation or ENSO events. These events are triggered by natural changes in sea temperatures and air pressures over the Pacific Ocean. Although ENSO events are a natural phenomenon, it has been predicted that global climate change may increase the frequency and intensity of these events. This is a serious concern for semi-arid countries, including South Africa, because most of the saline lakes are found in these areas, and the frequency, duration, and severity of droughts are predicted to increase greatly in these areas anyway. Overall, ecosystems that experience predictable or seasonal disturbances such as droughts and floods quite often are more resilient than their stable counterparts. The resilience and resistance of aquatic biota to disturbances are driven by their ability to either tolerate, acclimatize to, or avoid disturbances. These include being able to move away from the disturbance, go into a state of torpor or hibernation until the event has passed, or use physiological mechanisms to live through the disturbance. The time necessary for recovery to occur is a measure of the ecosystem's resilience. Recovery in aquatic invertebrate communities occurs through the hatching from resting egg stages in zooplankton, active dispersal from nearby refuges by aquatic insects, and the eventual passive dispersal of other aquatic invertebrates. The level of recovery of an ecosystem will depend on a number of factors, including the duration, intensity, and frequency with which the disturbance occurs. There are numerous lake ecosystems in South Africa, but research on their ecology, particularly regarding their aquatic fauna, is scarce and outdated. There is also limited information on how natural saline lakes respond to drought, and thus assessing such a system through a disturbance event is necessary to provide an indication of the community and ecosystem resilience in the face of climate change. The aim of this study was therefore to evaluate the effects of a superseasonal drought and accompanying changes in salinity on the aquatic invertebrate community of a naturally saline lake of South Africa. Our study took place on the lower Pongola River floodplain in the northern parts of KwaZulu-Natal, just below the Vanderkloof Dam near the town of Jozini. Here, located near Swaziland and bordering Mozambique, is the small and not so well known Ndumo Game Reserve. This is a rather small reserve of about only 11,000 hectares. It was established in 1924 with the aim of conserving hippopotamus and was proclaimed as a Ramsar site in 1997 because of its very rich diversity of terrestrial and aquatic habitats. It is also a biodiversity hotspot with some of the highest diversity of birds, reptiles, and amphibians. This game reserve also houses the third largest crocodile population in South Africa. Located within this reserve is Lake Inyamiti, the largest inland water body of the Ndumo Game Reserve. It is a naturally saline lake with an average salinity of about 3 grams per litre. The Pongola River floodplain is located on a marine Cretaceous bed which leads to naturally saline groundwater that seeps into the water table and Lake Inyamiti. The lake receives water in one of 3 ways: from annual or biannual controlled flood releases from the Vanderkloof Dam, through natural floods from the Estuaries River, which forms the border of the reserve with Mozambique, or through rainfall that occurs within its catchment. We sampled from 4 different sites around the lake, representing the 4 major habitat types around the lake, including open water, vegetated areas, and muddy and sandy substrates. This figure indicates the monthly flow of the lower Pongola River from 2010 to 2017. 2010 to 2014 is considered the period of pre-drought, as there was normal rainfall in the region and annual flood releases from the Vanderkloof Dam, represented by the high peaks on the graph. From 2015 to 2016, South Africa experienced the effects of an ENSO event that also caused a drought. This caused severe drought across the country, and as you can see, the lower Pongola River was not unaffected. Severely decreased rainfall occurred in the region, and there was no flood release from the dam since dam levels were below 30%. 2017 onwards is considered post-drought, as the worst of the ENSO event had passed. There was also increased rainfall in the study region, including around the lake, and small natural floods from the Estuaries River that allowed water inflow into Lake Inyamiti. Lake Inyamiti was sampled a total of 9 times, twice during pre-drought, 3 times during the drought, and 4 times during the post-drought. Without going into too much detail, we collected zooplankton and macroinvertebrates for biodiversity analysis. We measured in situ water quality parameters, and water was collected for further chemical nutrient analysis. For data analysis, diversity indices were calculated, including the usual suspects of Margolef species richness, Pelo's evenness, and the Shannon diversity. A CCA, or canonical correspondence analysis, was also created to look at the differences in biodiversity and water quality between the sampling periods. Okay, so what did we find? First, there was a severe increase in salinity from the pre-drought to the drought, increasing from about 4 grams per litre during the pre-drought to more than 10 grams per litre during the drought. At the same time, we saw a great decline in aquatic invertebrate diversity during this period. Aquatic invertebrate diversity went down by almost half, from 42 taxa to 23. As part of the larger project, we also monitored the fish populations of the lake and found that only one species of fish was able to survive during the severe drought, and that was Oreochromis mozambicus, the Mozambique tilapia. From the drought to the post-drought, salinity decreased by about half to approximately 5 grams per litre. Concurrently, there was an increase in the number of aquatic invertebrates, with 77 taxa being found, thus surpassing even the pre-drought. At the same time, there was also an increase in fish species from 1 to 9 species of fish. If we compare the pre-drought and post-drought with one another, we see that the biodiversity and the salinity of the lake started to return to what it had been before the drought, thus showing some form of recovery. The drought caused a severe increase in salinity and a subsequent decrease in biodiversity during the drought, as almost half of the taxa previously found were absent. The community structure was also altered, as only taxa that are known to be highly tolerant to drought and salinity were present during the drought. These included the copepod Harpacticoida and ostracods, both of which have life histories of living in more saline environments. Macroinvertebrates including Coryxidae and Hydrophilidae were also present, both of which are known to be able to tolerate highly saline conditions. With regard to fish, it is well known that Oreochromis mozambicus is well adapted to living in both fresh and estuarine habitats, thanks to their osmoregulating abilities, which is why it was the only species of fish able to survive in the lake during the drought. Taxa that were absent during this time included the mollusks Bolinus and Corbicula and the highly invasive snail species Terebia granifera. Interestingly, this particular snail species has been found in saline and estuarine conditions, but it would appear that even this species was unable to cope with the salinity of Lake Inyamiti during the drought. All other fish species known to occur in Lake Inyamiti, including tigerfish, sharp-toothed catfish, and many of the barbs, were also absent during the drought because of their inability to tolerate the high salinity levels. Salinity decreased as a result of freshwater inputs from the Lesotho River and isolated rainfall in Lake Inyamiti's catchment. As salinity decreased, there was an almost immediate increase in aquatic invertebrate biodiversity, as there were already more invertebrates at the beginning of 2017 than during the drought. There was rapid recolonization by aquatic invertebrates. This occurred through active dispersal by flying insects from nearby water sources and zooplankton that hatched from the sediments. This recolonization and the great increase in biodiversity was indicative of high productivity and a system in the process of recovery. Several taxa were still missing at the end of the study. However, they were not completely absent or extinct because they were still present in the nearby water bodies, including the Pongola and Lesotho rivers. These taxa, including snails and fish, are greatly limited by their dispersal mechanisms and thus would require a flood for them to be able to truly re-establish in Lake Inyamiti. The similarity between the pre-drought and post-drought indicated the resilience of the ecosystem and the limited impact that the drought had. This is related to the seasonal periods of drought and flood that Lake Inyamiti experiences under normal circumstances. Ecosystems that experience frequent bouts of disturbance are more resilient and inclined to rapid recovery, as the aquatic community is well adapted to overcome disturbances. The supraseasonal drought and greatly increased salinity resulted in a substantial decline in invertebrate species richness. The aquatic invertebrate community also changed, and the number of species declined due to their inability to resist and tolerate the greatly increased salinity. Only the most tolerant and halophilic biota were able to remain. Resilience of the lake was illustrated through the rapid recovery of the aquatic invertebrates following the drought. Biota with aerial dispersal capabilities and desiccation-resistant life stages in the sediments recolonized rapidly with the decrease in salinity. Other taxa that were still absent at the end of the study were largely limited by their dispersal mechanisms. Aquatic biota that live in Lake Inyamiti are clearly adapted to living in constantly changing conditions. This greatly assisted their ability to rapidly recover from the supraseasonal drought and increases in salinity. However, it must also be said that it is likely that more frequent and prolonged periods of high salinity conditions are still a major risk to the integrity of the lake, as there may come a time that the drought is so prolonged that not even the most tolerant taxa would be able to survive. It is also clear from the results that freshwater inputs into the lake are an integral part of sustaining the ecological integrity of Lake Inyamiti, and it is thus imperative that sustainable management strategies include actions that will prevent change or reduction in the water supply to this unique ecosystem. That brings me to the end of my talk. Thank you very much for your attention.
All right, thank you. Well, thank you guys for listening, actually. It's weird to talk about yourself. And if you have any questions, please feel free to ask me in the chat box. Our next speaker is Melissa Pollard, and she will be telling us more about her PhD work on the habitat use of marine fish.
Hey ladies and gentlemen, today I will be telling you about one of the aspects of my PhD project, namely microhabitat utilization by two common estuarine-associated juvenile fish species in a shallow sheltered rocky cove. Acoustic telemetry affords a unique opportunity to track the movements of individual fish in the wild and can provide fine-scale spatiotemporal data such as home range and the identification of movement corridors that are important for assessing the behavioral ecology and life history of species. Advances in tag miniaturization now allows for the tracking of early juveniles to assess the relative importance of nearshore nursery habitats for marine species and alternative nursery habitats for estuary-associated species. The aim of this part of the study is to assess the movement ecology of Cape Stumpnose and blacktail in a shallow rocky cove in Algoa Bay using acoustic telemetry. Larvae and juveniles of Cape Stumpnose are known to recruit into vegetated habitats of estuaries. Although Cape Stumpnose is thought to be estuarine dependent because food resources, mainly seagrass, are absent from the sea, red algae when washed into estuaries from the sea is often consumed in higher quantities than seagrass. This highlights the possibility that red algae may also provide food resources and shelter in the nearshore marine environment. Blacktail are primarily marine inshore species with juveniles utilizing tidal pools and inshore reefs as nursery areas. They are, however, also often associated with seagrass beds in warm temperate estuaries. This could indicate possible competition between these two species as they utilize the same environments and habitats as nursery areas. Flat Rocks is a sheltered shore located just inside of Algoa Bay. It has a gentle slope consisting of pebbles and boulders with sand patches occurring in between. There is an abundance of seaweed growing on reefs in the area, and shallow intertidal pools are also abundant. The wave action at Flat Rocks is light, making it a good environment to deploy acoustic telemetry receivers. We deployed 380 kHz receivers in the small bay and one in an adjacent smaller bay. The receivers were deployed in different habitats that include sand, low-profile reef, and high-profile reef. A fifth receiver was deployed in a deeper reef to see where the fish move out of the bay into deeper waters. Temperature loggers were attached to the moorings, and one aqua measure that records dissolved oxygen and temperature was added to a mooring in the bay. Fish were caught using rods during the months of August to October. 15 blacktail ranging in size from 95 to 298 millimeters total length, and 5 Cape Stumpnose ranging in size from 202 to 227 millimeters total length were caught during this period. Once fish were captured, they were anesthetized using 2-phenoxyethanol at a concentration of 0.4 milliliters per liter of water. Once anesthetized, fish were operated on to place the telemetry tag inside the body cavity, and 2 silk sutures were used to close the incision. The fish were then placed in a recovery bath and monitored to ensure full recovery before being released back into the bay. Data downloads have occurred during spring low tides over the last 3 months. The download procedure includes 2 divers swimming out to the moorings and retrieving the receivers. The data is then downloaded onto a laptop and the receivers are cleaned and reinitialized before being returned to the respective moorings. The first download produced over 76,000 detections, the second over 207,000 detections, and the third over 280,000 detections. After 3 months, there has been over 571,000 detections between the 5 receivers. Preliminary results indicate high residency by the tagged fish to the bay. While some fish have disappeared from detection range, most fish stay close to the bay. Most detections are seen on the high-profile reef, shown in blue, followed by the low-profile reef, shown in green. Some fish have ventured out of the bay and have been detected in the small adjacent bay, shown in orange. Our biggest Cape stumpnose of 227 mm is often detected in the smaller bay. The smaller fish tagged, seen at the bottom of the plot, seem to prefer the high-profile reef in the bay. Moving forward, another data download will occur before removing the receivers from the bay. The data will then be analyzed and environmental data will be added to see what factors play a role in the movement of Cape stumpnose and blacktail in the shallow coastal bay. Thank you to all the fishermen and my tagging team for all their help. Thank you to Innova Sea for the use of their receivers during this study.
Great, thank you so much, Melissa. Our next presentation is by Marlies Treater, but before we get to that, I just want to mention that once the session is over, a poll will appear for people to vote for their favorite MSc and PhD presentations. Please go vote for your favorite presentation before you leave for lunch. Now we have Marlies, and she will be telling us about the parasite diversity of catfish in South Africa.
Hello, my name is Marlies, and I will be presenting on the parasite communities of 2 invasive populations of the African sharp-toothed catfish, Clarias haripinus, in South Africa. The African sharp-toothed catfish naturally occur in freshwater systems of South Africa, with the Orange River in the Northern Cape and the Mtamvuna River on the east coast of the country representing their southernmost distribution in Africa. However, several factors contributed to its distribution beyond these rivers in the country through several pathways. From literature, it is clear that parasitic community data of the catfish is primarily available from dams and rivers in the provinces where the main freshwater parasitology research groups are active, here highlighted in dark grey. No studies have been conducted on the host in systems in the southern provinces of our country. Thus, the aim of this study was to investigate the parasite communities of invasive populations of Clarias haripinus in the Eastern and Western Cape. Hosts were collected from the Great Fish River and Riviersonderend in the Eastern and Western Cape respectively using various collection methods. After euthanasia, each individual was screened for metazoan parasites on the skin, fins, gills, and in all internal organs. All parasites found were fixed according to standard protocols for each taxon for later morphological and molecular identification. Catfish from both rivers were infected with ectoparasites on the gills and endoparasites in the cranial cavity, visceral cavity, and organs. 91% of catfish from the Great Fish River and all individuals from Riviersonderend were infected with any combination of parasites and a similar intensity of infection. All individuals from the Great Fish River were parasitized in the gills with species of Quadracanthus. 4 known species of the genus and a 5th unknown species, Quadracanthus sph, were present. Only 3 species of Quadracanthus were found from the gills of hosts in Riviersonderend. These species, except Quadracanthus sph, are all known from the catfish in its native distribution range in South Africa and Sudan. Catfish from the Great Fish River were parasitized with adult Orientocranium batrachoides in the intestine, whereas Metacercaria of Tiludalphus machinensis were found to parasitize the cranial cavity of 27% of the catfish. No infections with Digenea were found in the hosts from Riviersonderend. Nematodes infected catfish from both localities. Larval individuals of the generalist Contrasecum were found in the visceral cavity in hosts from the Great Fish River, and adults of Paracamelanus ceatopharynx were found from the intestine of host individuals from Rivier Sonder End. Hosts from Rivier Sonder End were also parasitized by two cestodes, Parasonella lafoni and Tetracampus celioteca. Both species are known from the catfish in its native regions of South Africa, but this is a first for the Western Cape. Catfish from the Great Fish River were not infected with any cestodes. Lastly, a species of the copepod Ergacillus were found from the gills of Clarias gariepinus at both localities. This is the first study to present parasitic community data from invasive catfish populations in South Africa. It also highlights the void and disparity in our knowledge on the distribution of this host and its parasites in our country. All the parasite species found during this study are well known or specialists of the catfish, meaning that all these parasites were co-introduced with the host into the Great Fish River and Rivier Sonder End, and also represents new distribution records. Parasites not commonly associated with Clarias gariepinus were absent, indicating that to date no parasite spillback from native hosts or other invasive species has occurred. It is also clear that some parasite species did not succeed during their translocation into these systems, as only 9 and 7 taxa are present, whereas between 11 and 32 have been reported from source populations, as well as single populations in the host's native distribution range in southern Africa. This supports the enemy release hypothesis. This decline in species richness can be attributed to environmental barriers, the natural absence of these species from source populations, or simply the absence of suitable intermediate hosts. Future research should focus on elucidating the parasitic communities of not just the catfish and their relationships, but also endemic and native species to assess local diversity and the potential for spillover or amplification. There is very little to no records or a so-called parasite library for many freshwater fishes from South Africa, especially in regions such as the Western and Eastern Cape that holds unique fish communities. Dedicated or collaborative research between relevant stakeholders would aid in our understanding of these simple yet complex organisms.
Great. Thank you, Marlies. I always find your talks really interesting and just a little bit gross as well. Next, we have Vuyo, and she will be telling us about the migration of leervis in South Africa.
Hello everyone, my name is Vuyo Tumko. I'm an MSc student at SAIAB. As part of my MSc, we wanted to understand migrations of leervis along the South African coastline using acoustic telemetry. The project was supervised by Professor Cowley, Dr. Murray, and Mr. Parkinson. Leervis is a popular recreational fish species in South Africa, and this is due to its large size. They can reach a maximum length of 180 centimeters and can weigh up to 32 kilograms. In South Africa, they are distributed from the Orange River to KwaZulu-Natal, and they are SASE red listed. That is, no sale of the species is allowed anywhere in South Africa. This is due to the collapsed fish stocks caused by heavy fishing pressure on the adult spawning in KwaZulu-Natal. The mark and recapture methods have shown the species to take seasonal migrations from Eastern Cape to KwaZulu-Natal, but it remains uncertain whether the leervis adopt partial migration strategy, whereby not all individuals migrate annually, or whether migrating individuals return to specific sites. From the study, we wanted to find out why are leervis migrating, which life history stages are migrating, when are they migrating, where are they migrating from and to, what do they do after migrating, and what does this mean for the conservation of the species? To answer these questions, passive acoustic telemetry methods were used. In passive acoustic telemetry, a fish is tagged with a uniquely coded transmitter that releases signals. These signals are then decoded by fixed acoustic receivers. The data on detection of the species is then collected from those receivers. For this specific study, we had 71 leervis that were monitored from 2011 to 2020. And from that, we got 748,649 valid detections, which were then analyzed. From the study, we were able to discover that fish from all areas, that is Eastern Cape and Western Cape, migrate to KwaZulu-Natal in winter and return to Eastern Cape in summer. And that migrations are influenced by the life history stage of a fish. That is mainly adults and some subadults migrated, which suggested that migration could be driven by spawning, since at that size, a fish is presumed to be sexually matured. Another important finding was that the timing of migration is regular, occurring from June to November on each year. From the graphs, you can see fish departed Eastern Cape in June, arriving in KZN between June and July, and a return migration from KZN in October to Eastern Cape in November. We were also able to show partial migration, whereby some individuals migrated and some remained resident to their tagging locations. Fish also displayed a homing behavior by returning to their tagging locations and surroundings, and we assume the reason behind this behavior was feeding in familiar environments, where they will replenish energy stores and build up gametes for the next season. From the study, we were able to show that fish from all areas migrate to KwaZulu-Natal and return to predictable locations. And also, acoustic telemetry was able to show evidence of partial migration. For this reason, information on partial migration and phylopatry needs to be incorporated in conservation planning strategies in order to conserve the species. Thank you.
Great, thank you very much for that. Next we have Philantle, who will speak about her work on the development of eDNA protocols.
Greetings everyone, my name is Philantle Nyawo, and I will be presenting on the protocol development for assessing marine fish assemblages using environmental DNA. Environmental DNA sampling, which refers to the capture of DNA released by organisms in the environment, has emerged as a new approach for assessing biodiversity in the marine environment. Compared to traditional monitoring techniques, eDNA is emerging as a non-invasive biodiversity monitoring tool that is also promising to be time and cost effective. As a new technique, eDNA sampling methodologies are very diverse and are under continuous development. Having a precise methodology chosen when sampling eDNA is very important, as this will play a major role in the detection of species or community being investigated. A great number of different protocols have been developed to collect, extract, and analyze eDNA in the marine environment. However, the method still lacks field implementation to evaluate its effectiveness and applicability as a marine biodiversity monitoring tool in the South African context. Which then brings us to the aim of this study, which was to develop and standardize a protocol for assessing marine fish assemblage structure in offshore reefs using eDNA. Our objectives were to determine the optimal volume to be collected per sample, evaluate suitable stabilization buffers for preserving the samples in Stereovex filters, and conduct a direct comparison between eDNA and StereoBrav results in terms of family or species composition. Sampling was conducted in the Titicama National Park Marine Protected Area in both the shallow and deep reef sites. For environmental DNA, samples were filtered through Stereovex filters and preserved in 2 mils of long Maya or TL buffer. DNA extraction conducted in a designated eDNA lab was done with the eZNA extraction kit. PCR was conducted on 3 replicates of each sample extracted, and purification of the pooled replicate was done using the MAC-bind total magnetic beads. StereoBrav videos were analyzed using the Event Measure software, and all fish identified to species level. When looking at the total number of reeds detected per sample volume, preliminary results showed that 0.5 liter detected a significantly lower number of reeds, and higher number of reeds were detected in the 3 liter samples, which was the highest volume investigated. A similar pattern was observed when looking at the total number of species detected per sample volume, indicating that the higher the number of reeds detected, the higher the number of species observed. When looking at how stable the DNA was in the different preservation buffers, more reeds were obtained in the samples that were preserved with Longmire's compared to samples in TL buffer, suggesting that Longmire's worked slightly better than TL buffer. TL buffer was what was suggested by a lot of literature, while Longmire's was what we were testing out in this study. When comparing eDNA to stereographs and summarizing the species composition between the two methodologies, we found that eDNA detected reeds belonging to 23 families, while stereographs identified fish belonging to 15 families. The most diverse family included sparrows, with 13 species detected when using eDNA and 16 species identified in stereograph videos. For both methodologies, carpenter was in the top 3 species that were abundant in the study. Still looking at the composition between the two methodologies, some species such as the black mussel cracker and Dane sea bream were detected using environmental DNA and were not seen in stereograph videos. Some species were identified in stereograph videos and were not detected by eDNA, and these included the brown shy shark and leopard cat shark. Species including red Roman, carpenter, and smooth-howled shark were detected by both methods. It is important to note that some of the species recorded in the stereographs were not part of the reference sequence library that is still in progress, and that could be a reason why they were not detected by eDNA. Once the reference library is complete, we will then reanalyze the data. In conclusion, we found 3 liters to be the optimal water volume, reported that Longmire's are slightly better than TL buffer, and both methodologies are complementary. For example, a large number of species was detected by both stereographs and eDNA. Thank you.
Thank you very much for that talk. Next, we have Daniel van Blerk, who will be telling us about his MSc work on the effects of invasive fish on frogs.
Hello everyone, my name is Daniel van Blerk. I am a master's student at Stellenbosch University and I'll be presenting the first chapter of my thesis today. A global review of invasive species impacting amphibians identified invertebrates as the most consistently negative impacting taxa. This is largely attributable to the invasion of freshwater crayfish species. Although these crayfish are highly impactful, alien fish have a richer history of introductions across more established species and with a wider invasive range, including countries like South Africa. A review of alien fish in South Africa found that 55 non-native fish species have been introduced to the country, with at least 44 successfully establishing. Sport fishing remains the leading contributor to continuous introductions, with most introductions occurring in fishless streams. Rainbow trout and brown trout are introduced to the upper reaches, and further downstream, where the water is warmer and habitat no longer suits the persistence of trout, smallmouth bass are introduced, with largemouth being introduced to the lowest reaches and catchments. Many South African streams are thus under intensive stocking regimes, leading to highly impactful fish species now occupying the same habitat as native stream amphibians. 2 studies have looked at the impact of invasive fish on ghost frogs. Their results show a significant decrease in tadpole abundance below waterfalls. Waterfalls are dispersal barriers to fish, making for an easier comparison between sites within a stream. However, not all streams have natural dispersal barriers, and deliberate introductions will occur above these dispersal barriers. Therefore, a more comprehensive study will determine whether the impacts reported by Avedon et al. And Casing et al. Are reflected across multiple streams, ghost frog species, and invasive fish species. The aim of this study is to determine the impact of invasive fish on ghost frog tadpoles. To achieve this, I will measure tadpole densities in the presence and absence of invasive fish whilst accounting for environmental variables. I predict that ghost frog tadpole densities will decrease in the presence of invasive fish due to predation by the invasive fish. The results of the study could benefit conservation planning regarding ghost frogs, as well as aid in management actions surrounding invasive species control. So onto materials and methods. Ghost frogs are known as the family Heliophrinidae. They consist of 7 species found along the escarpment from the Western Cape to KwaZulu-Natal. Tadpoles spend up to 2 years in the water and are morphologically adapted to the fast flow of permanent mountain streams. This exceptionally long development time is attributed to the low water temperature, which is a key habitat predictor for ghost frog tadpoles, along with flow, pH, substrate and substrate type. Due to the specialist habitat requirements, Hewitt ghost frog and Table Mountain ghost frog are listed by the IUCN as endangered and critically endangered respectively. To determine where I'll do fieldwork, I used ghost frog occurrence data obtained from the SAIAB and loaded it onto a Google My Maps. I then used occurrence data downloaded from FBIS for rainbow trout and smallmouth bass and layered that onto a map. I chose prospective sites based on 10 streams where frogs and fish occur and 10 streams where only frogs occur. Tadpole sampling will consist of manual searching in timed intervals. I'll start as high up in the stream as possible and move down 20 meters. 20 minutes will be timed for searching the substrate and collecting tadpoles in a kick net. After 20 minutes, the tadpoles are photographed on a calibrated measuring tray and released. The number of tadpoles collected in 20 minutes over the area covered in the 20 minutes will be used as a metric for tadpole density. This method follows ongoing research done on Heliophrine rosae on Table Mountain. Important habitat variables will be measured with a HANA water quality multimeter, and physical properties of the stream will be measured using a tape measure and calibrated depth rod. Electrofishing will be used to sample fish. 20-minute searches with the electrofisher will be conducted with all fish collected in a keep net. Fish will be photographed on a calibrated measuring board, identified to species level, and released unless specified otherwise by landowners or governing bodies. I will use the CCA package in R to run a canonical correlative analysis. This is gaining popularity in aquatic studies due to the researcher being able to include qualitative and quantitative environmental data. I will include all the data related to fish as environmental variables along with the habitat variables, as the observed tadpole density could be influenced by something other than invasive fish. I'd like to thank all the contributors to my master's thesis, and lastly, I thank you all for your time today and welcoming.
Thank you very much, Daniel. That brings us to our final speaker for the session, that is Nobuhle Mpanza, who will be talking about her MSc work on Oreochromis mozambicus from the Sundays River. Yes.
Good day, everyone. My name is Nobuhle Mpanza, and my talk today is on the age and growth of Oreochromis mozambicus and the Sundays River catchment in the Eastern Cape. So freshwater ecosystems are under pressure from a number of stresses, one of which is the continued introduction of invasive species that threaten both the biodiversity and integrity of these ecosystems. One such species is Oreochromis niloticus that was first introduced in South Africa for aquacultural purposes in the Limpopo River and has since escaped and established viable feral populations. These newly established populations of Oreochromis niloticus have been implicated as having adverse impacts on receiving river systems, such as decreasing endemic fish abundances and the local extinction of native congenerics through competitive exclusion and hybridization. One of the native congenerics identified by NEMBA as being at the risk of extinction is Oreochromis mozambicus, a native Southern African cichlid closely related to Oreochromis niloticus. So proper assessment and management of fisheries requires an understanding of life history parameters of a species such as its age and growth. Hence, the knowledge of age is crucial in estimations of growth, maturity, as well as its mortality rates. Therefore, age and growth studies are important in determining the impacts of exploitation on fish stocks, as well as improving our understanding of factors affecting recruitment processes. For this study, we used sagittal otoliths as they are able to record the entire lifespan of species. So the age and growth information from this study will assist in assessing and predicting potential impacts of Orochromis niloticus in the Eastern Cape region if it was to be introduced in this region. So the main aim of this study was to determine age estimates of Orochromis mozambicus using section sagittal otoliths in order to provide growth rates for the population, and then make comparisons with that of Orochromis niloticus based on published data. And then the specimen from this study were collected from the Emil Swart Pond and the Sundays River catchment. A specimen were measured for total length, weighed, dissected, and sexed, and then the otoliths were extracted in order to conduct the age estimation study. And then all analyses were done using R and Microsoft Excel. And then for our results, firstly, we found that the oldest female was 6 years of age with a total length of 288 millimeter, and the males range between 1 to 6 years of age, with an exception of 2 specimens, which one was 12 and the other was 15. And then the largest male was 297 millimeters. The juveniles were either 1 or 0 in age, ranging between 32 millimeters to 83 millimeters in total length. The land-weight relationship showed an increase in weight with an increase in total length. The model showed a good fit on the transform data, with the goodness of fit indicated by R squared is equal to 0.944, and the growth exponent parameter B was equal to 2.688, showing an allometric growth for Orochromis mozambicus. And then when we look at the growth model for combined sexes, it was best described with an asymptotic length of 322.5 millimeters, a growth coefficient of minus 0.201, and the age estimated 0 of 1.027. The population had relatively fast growth for the first 4 years, and thereafter slowed down with an increase in age. And then comparisons with Knight Tilapia were based on literature, and they showed that Knight Tilapia had higher asymptotic length than Orochromis mozambicus for combined sexes. And when we look at sex-specific differences for both species, males were found to grow faster than females. The growth model provided a good fit to landed age data for Orochromis mozambicus. Growth increased rapidly, followed by a reduced growth, then coincided with the species approaching its first maturity. Growth patterns in cichlids are consistent with a decrease in somatic growth as fish allocates more energy to gonadal growth as they approach sexual maturity. The sex-specific differences, on the other hand, in asymptotic length may be attributed to sexual selection for larger, potentially faster-growing males because of their aggressive territorial behavior during spawning. And then the asymptotic length and growth coefficient for Orochromis mozambicus in the present study was lower compared to that of Orochromis niloticus. Hence, Orochromis niloticus had a faster growth than Orochromis mozambicus. Furthermore, Orochromis niloticus is known to utilize a wide range of food resources, which may be a factor in their faster growth rates. The dietary overlap between these two species could result in the displacement of Orochromis mozambicus in the Eastern Cape if Orochromis niloticus was to be introduced in this region. Currently, there is a lot of published data on the distribution of Orochromis niloticus in South Africa, as well as a lot of restriction put in place by NEMBA, yet there is a lack of published literature on its actual impact it has on receiving rivers, as well as its closely related congenerics. The study then forms the basis in predicting the potential impacts Knight Tilapia has on its closely related congenerics such as Orochromis mozambicus. Thank you.
Thank you so much for that talk. That brings us to the end of our session. Please vote for your favorite presentation before you leave for lunch, your favorite MSc and PhD presentations. Thank you very much to all our speakers for their presentations, and thank you for joining the session. Please enjoy lunch and join us again at 2:30 for the next session.